All right, welcome back, and this is time to start the last lecture of the morning session. So this is again the last lecture of the pedagogical series of lectures, and this is will be taken by Professor.
Thank you very much. So, in this last part of my talk, I'd like to explain our recent research effort on the genome physics. So, in particularly that our efforts are somehow similar to the Kian's talk, the last speaker, meaning that we are considering about the coarse graining and the phase separation. But we'll talk about things in different spatial and time scales. And my talk consists of these parts. The first, I'll start from the introduction, and then I'll talk about compartments, chromatin compartments, and then talk about chromatin domains, and I talk about the entire genome architecture. Then we discuss about dynamics or something like that. And everybody knows this molecule. And DNA is an acid, which means that there are many phosphate groups along the backbone chain. And these are bearing negative charges in usual conditions. So DNA is a molecule with plenty of negative charges. So in eukaryotic cells, DNA wraps around histone octamers. So histones here has positive charged residues, lysine or arginine along this side. And in the schematic writing, so histone has positive charge here and a negative charge here. So negatively charged DNA, winds around this side, and its N-terminal tail is disordered like this, and lots of positively charged residues are here. Many lysine residues are contained. So this positively charged tails usually wraps, entangled around DNA. Interacting together with the negative charge of the DNA. But sometimes it dissociates to interact with the neighboring nucleosome in this way. So in principle, these nucleosomes are, having the attractive interactions in general. But, this is the configuration dependent. So this is not a simple two-body attractive force like Coulomb interaction, or, but it depends on the configuration or some stochastic environmental conditions, something like that. Anyway, this is a structural unit of chromatin. So DNA wraps around almost 2 turns around histone octamers. And this is an illustrative scheme of how genome is organized. So this particle, histone octamers and wrapping DNA, is called nucleosome. And the array of nucleosome is called chromatin chain or chromatin fiber. Chromatin means the state of DNA bearing a lot of associating proteins. So, and this chromatin chain often forms loops of the size kilobase to megabase. And in particular, that the loops of the size of 100 kilobase or megabase is called topologically associating domains or chromatin domains. And these chromatin domains tend to gather together to form compartments. And inside the nucleus, different chromosomes do not mix greatly, but occupy each own territory. So this is a hierarchical architectural structure. And important question is how the these structure formation or the structure changing is correlated with the functional activity of cells, like transcription or the duplication. And this modern view was established through the recent advancement in the measurement technologies, including the super resolution microscopy. Super resolution microscopy is the microscopic technique to see the tiny things smaller than the length of the optical wavelengths. And particularly that high throughput biochemical analysis, including IISc, was very contributed greatly to reveal these structures. And we should say that we need to develop computational models to understand these different experimental data in a unified way. So to do that, we need to understand the physical principle of this genome organization. Just briefly explain what is IISc. And IISc is probably coming from the name high throughput chromosome conformation capture. And this was invented by this group in about 10 years ago. So the first cells were fixed with formaldehyde, formalin. Formaldehyde was added to cells. Then that the cell will be killed. The cell dies, and because that proteins inside cells denaturated, and in particular that abundant proteins near DNA was deformed to bundle two different DNA chains. Then that some enzymes were added to cut these sides. For example, by adding Hindi 3, many positions, approximately each 1 kilobase, 1,000 nucleotide pair region is cut with this enzyme. So we have a lot of fragments like this. Then that some end marks were added to this end and ligated again to form a ring. And then this tiny ring was, these bounded proteins were eliminated by deleting formaldehyde from the solution, and with the, for example, that ultrasonic sonification, that this DNA is physically cut it with the short fragment, and we obtained this kind of units. genome-wide. And by reading this sequence in next generation sequencer, then we can find which part was originally in the physical vicinity, which part was in contact in original cells. So this is the technique of IISc, and doing this genome-wide, so many read pairs were obtained, 1.1 billion reads in the more recent experimental data with the 1 kilobase resolution. So this is a typical results of the IISc measurement. This is the case for the chromosome 14 of human cells, and this pattern shows the frequency of how different positions in chromosomes are in the physical vicinity before adding the formaldehyde. I mean that this is not a single cell measurement, but a colony of cells were treated and measured simultaneously, that it contains about 1 million cells. So this is an average contact number in a colony, or that a kind of the probability to find contact, physical contact between different chromatin sites in these chromosomes. So this gives information about the 3D structure of the genome. And the many computational efforts were made to generate 3D chromosome conformation starting from this IISc data. But probably that chromosomes in individual cells largely fluctuate. So, this information does not determine a single structure. I mean, the situation is somehow similar to the NMR measurement determination of the protein structure. But in the protein case, that protein conformation is fluctuating around the native structure. But in this case, very different chromosome structures are superposed in this information. So it is not straightforward to determine the single representative conformations from this IISc data map. But anyway, it is possible to do that with some method like maximum entropy method to take into account this information map.
Just one question.
Yes.
So in this kind of IC map, what do we have on the two axes? I mean, the nucleotide sequence, or what do we?
This is the sequence.
Okay.
This is the same sequence, I mean. Yeah, so that the sequence position here and here has this probability, for example. This position and this position has the contact frequency of this, color intensity or something.
There are also contact maps for, I mean, you can also have another kind of contact map where you can have all chromosomes.
Right. Genome-wide. So we can have the inter-chromosome contact map. That is much thinner. I mean that intra-chromosome contact is much thicker patterns. But we have the genome-wide, the big matrix data in this way. I mean that because with the 1 kilobase resolution, the human genome has 3 gigabase size, which means, and with the 1 kilobase resolution, it has the a few million, dots along this line. So, the whole data is approximately with this size. So this is like this. So this big information, I mean, was treated to construct a computational model of chromosomes. Yes. Right, right. So in real cells, so for human cases, that it has 46 chromosomes, 23 doubled chromosomes, 23 pairs of chromosomes. So that interactions among chromosomes are an important point. But the first, around 2017 or 2018, individual chromosomes were first computationally modeled. So, I mean that we should have the whole genome model to resolve that problem. So that is our motivation to start this project. And what we learn from this IC contact map is that block pattern along near the diagonal line and the checkerboard pattern in the off-diagonal region. So block pattern implies some domain formation. So this pattern, this is this from this to this is 100 megabase. So each block pattern represents several megabase or 10 megabase. This is a big domain. And by further, looking at this point, this is 10 megabase length. So each block appearing here has several hundred kilobase or 1 megabase size. And this is called topologically associating domains. And by further enlarging this part, we have this typical pattern. We see dots at the corner and the stripes inside the block. So these, for example, intra-domain, this is the domain. This is one domain, one tad. And inside tad, we have stripes and peak. So this should represent some functional complex of chromatin, like enhancer chromatin, enhancer promoter loop or something like that, or super enhancer hub. Is shown, enforced pattern inside the domain. And these dots, the corner dot, shows this domain, for example, is a loop. And the contact between the loop boundary can be seen as a dotted peak like this. And people believe the cohesin, this is a molecular motor, is responsible for this looping. The cohesin is a ring-like molecule. It has a big ring. Like this. So, and there are several subunits associating this to load the cohesin on the DNA or unload the cohesin from the DNA. But in this big ring, DNA can, penetrate. And possible explanation is that the two chains can penetrate. So this cohesin ring can bundle the chromatin chain. Cohesin usually appears in the S phase, in the timing of the DNA chromosome replication. So to bundle the sister chromatoids, you mean the chromosome just after replication can must stay near to each other. So that is this cohesin binding bundling is responsible for that process. But so this is essential for the cell cycle progress. Without cohesin, the cell will die. But in the interface, before the replication, cohesin can bundle the same chain like this. And there's a lot of argument. But at least this cohesin will diffusively move along the chain. And if this cohesin moves in a somewhat biased way by consuming ATP, this subunit consumes ATP. This is the hypothesis, the so-called loop extrusion hypothesis. So the relatively movement of the chromatin chain is to extrude the loop. I think there are some arguments, who do not agree with this hypothesis, but many experimental evidence shows that the cohesin really slides along the chromatin. And with this, chromatin loop extension, and this part becomes bigger, but this cohesin binds CTCF, and that stops the sliding motion, and so that this CTCF binding loci becomes the loop boundary. Actually, that the CTCF binding is often found in this peak position. So this is a basic picture for the loops and domains. And these domains may collapse to various extent through the nucleosome interactions. But these interactions depend on how histones are modified, or saturated or methylated. So that depends on the functional activity of that domains. In the transcriptionally active domains are often accentuated, and accentuation provides the minus charge to histones, so that the repulsive interactions become the loop more extended. But an interesting thing is that different types of domains, there are many efforts to classify domains, but roughly speaking, we have two types of domains, type A and type B. So the name A and B was named in the 2009 paper of the Hayashi invention, but anyway, that people usually use this naming. So the transcriptionally active, rather open chromatin domain is type A, and transcriptionally inactive, chromatin in the heterochromatin region is type B, and type A domains tend to gather together to form compartment A, the larger structure. And the type B domains tend to gather together to form compartment B, like this. So in this illustration, the compartment B is red colored and accumulates around the nuclear membrane or around the nucleolus. Nucleolus is a liquid droplet, formed by the liquid-liquid phase separations. It consists of the ribosomal RNA and the related factors. So anyway, the transcriptionally inactive compartment B accumulates around the nuclear periphery or nucleolus. And the core region is occupied by gene-active compartment A. So this separation, it can be seen as a checkerboard pattern like this. So for example, with this picture, this domain and this domain belongs to the same compartment, but this domain and this domain, belongs to the different compartment. So the checkerboard pattern is a hallmark of compartmentalization. So we can say that these things, groups, domains, and compartments are the building blocks of the genome. So the questions, how these groups and domains are formed, and how their physical properties were determined, and how domains form compartments, and how these structures are related to the functions. So these are the essential questions in cell biology. And the many different hypotheses were arguing. So that is a fun that I try to convey the fun atmosphere of the scientific debates. I mean that things are not settled, I believe.
I have a question.
Yes.
So along with cohesions, there is also something called condenses which are involved in compaction of DNA. Do they play any role here? Condenses?
Condensing.
Yeah. So,
condensing works in the mitotic phase.
okay, not here.
But the condensing and cohesing are very similar proteins. So so this is called in a group of the SMC proteins, but works at a different timing in the cell cycle. So this is the live cell microscopy imaging measured by my Shima's group in Mishima. With this, the photoactivatable chromophore, M-cherry, is fused with one kind of histone, H2B. And then with the laser emission, chromophore were photoactivated. Then they can emit the fluorescent light to show these bright dots. But because the laser excitation at the first stage is weak in this case, only as randomly selected positions are photoactivated to allow the fluorescence emission. So we can distinguish individual single nucleosome movement in this picture, because that lots of densely distributed nucleosomes are in the nucleus. We cannot see all the nucleosomes. So only the very few randomly selected nucleosomes are bright in this picture. And so we can trace the movement trajectory of individual nucleosome genome-wide. And here that I like to emphasize that genome is very soft, flexible, always fluctuating. Yes.
So this is only a slight in confocal, image.
Yeah. So the microscope focus plane was very thin, probably a few hundred nanometer or so, with a 10 micrometer diameter nucleus. So this is only a intersection of the nucleus. So by changing the focal plane, we can see the nucleosome movement near the nuclear membrane or near the nuclear center, so that near the nuclear membrane, the movement is much slower.
Exactly what microscopic technique has been used for confocal or GIC?
I'm not. I'm not. Is it really confocal or not? I don't know. But one important technique is the fluorescence excitation is cast from the shunted angle from the slide cover. So only the surface was excited. Not the bulk nucleus was excited, but only the part near the slide was excited. So this is one technique to see the this delicate structure. Anyway, that I want to emphasize that genome is a soft matter, not a solid state matter. This is a plot for how each nucleosome moves. This is a distribution of the MSD, mean square displacement. This is distribution. So the red is the original histogram, and black is a smoothed curve using some Bayesian static technique. And it shows, this is a case of HeLa cell. We see the two components, fast moving chromosome and chromatin, and slowly moving chromatin with different exponent of MSD as a function of time. Anyway, that this should correspond to the hypothetical thinking is that this should correspond to the compartment A and the compartment B. Because we have two types of chromatin domains, it should show different movement. Yes. M star, this is a superposition of the 20 cells. And so for each cell, the average activity is different. That is one reason is that the biochemical activity is different among different cells, and the other is that the different plane was focused by the microscopy. Anyway, that the average position of the distribution is different from cell to cell, but bimodal feature is the same. So M star here is this bottom position. So by aligning different distribution on this point, then that many curves were superposed here, showing that different cells have qualitatively similar bimodal distributions. Pardon? So that we tried the Gaussian decomposition of this bimodal peaks. Yes.
So their single nucleosome is tracked. I mean, and their velocities are noted. Single nucleosomes are tracked and their velocities are noted. I mean, how they are moving. Right.
How they are moving.
Yeah. I mean, what is the speed of the nucleosomes by image analysis?
So this is the 0.5 sec duration, and within 0.5 sec, it moves a few nanometer. So this is around the domain size, and with less than 1 second, that the nucleosome moves around the domain. Of course, that in a longer time, probably that the more long wavelength movement, like hydrodynamical movement, would be possible. But with the the strong laser emission will damage the cells, so that we here plotted only the short time, around 1 second data. But anyway, that with the emphasis on the dynamic movement of chromatin, this compartmentalization, compartment formation could be phase separation. This is a microscope image of HeLa cell. This has a big nucleus. And here, HC, dark region, is heterochromatin region. So it has dense DNA. So it was shown in this picture that as a dark regions. And light gray region is a euchromatin region. And it has a less denser DNA packing. And euchromatin region is gene active, and heterochromatin region is gene inactive. Often the repetitive retroboson sequences are found in heterochromatin region. And so the terminology of euchromatin or heterochromatin came from the microscopy observation. And the terminology of compartment A and compartment B came from the high-C measurements. Then probably this corresponds to like this. So compartment A is euchromatin and compartment B is heterochromatin. And NO here is a nucleolus. So this is a liquid droplet of ribosomal RNA and other factors. So anyway, this looks like a phase-separated pattern of oil and water. In this case, that the oil glass interface energy is lower than the oil-water interface, so that the oil accumulates around the rim of this pellet. But anyway, we can imagine that some phase separation of different chromosomes or different chromosome domains generate this checkerboard pattern or this pattern. Because this is intimately related to the transcription activity or the replication timing, so it is also an important biological question, how this compartmentalization was happened. And so to support one idea to support this phase separation mechanism is the finding that HP1 forms a liquid droplet in vitro. But this observation was done in a rather extreme condition of the unphysiological pH. But anyway, it can form liquid droplets like this. And if the similar droplets are formed in vivo, then that chromatin will be gathered to form heterochromatin. This is one hypothesis. Because this is an HP1 structure. HP1 has the two domains, chromodomain and chromoshadow domain. And chromodomain binds to the H3K9 methylated sites. I mean, this is the 9th lysine of histone 3. Is when they are methylated, then that this chromodomain recognizes this mark and binds on the histone nucleosome like this. And chromoshadow domain can form dimer. So this can bridge two H3K9 methylated nucleosomes. So this should work as an effective attractive force between different nucleosomes. And when HP1 forms droplet, so it has an attractive force to gather many H3K9 methylated nucleosomes, then form the heterochromatin. But because that the H3K9 methylated histones are the frequent in the gene inactive region or heterochromatin. So this is one hypothesis, how the heterochromatin region or compartment B is formed. So HP1 mediated effective attraction will gather the nucleosomes. But I mean, with this hypothesis, two subsequent papers appeared in Nature in the same issue with this hypothesis, so that the people are very excited about that. And so many computational models were developed by using this assumption. So by assuming the attractive coarse-grained effective interaction between heterochromatin-like region, type B domains. But we can do the simulations with this assumption for a single chromosome. But when we simulate the entire whole genome, we have a problem. Because with the effective attraction between heterochromatin type B domains aggregate toward the center of the nucleus. This is like simulation of the planet formation. I mean that the heavy matters show the strong gravity and sink into the core of the planet. The same things happens here in the computational model. But the actual distribution is different. Heterochromatin is found at around the periphery of the nucleus. So this is the opposite trend. So people resolved this contradiction by assuming two competitive types of attractive force. One is the attractive force between type B domains, as we discussed. And the other is the attractive force between type B domain and the nuclear lamina. Nuclear lamina is a protein mesh located inside, covered inside the nuclear membrane. So if there is an attractive force between nuclear lamina and type B domain, and if we have achieved balance between them, then we can reproduce this observed structure. So this is the most widely believed hypothesis at present. People say like this in introduction or review papers. But I feel that it has some difficulty. One is that if we do that really in the computational models, fine-tuning of parameters are necessary between two competitive attractive forces. That's something strange to me. I mean that the mesoscopic structure, like genome architecture, should be robust against small perturbations. So that the necessity of the fine-tuning feels somewhat strange. Is one thing. And second is that the attractive force between type B domains and lamina is mediated by the protein like LBR. There are several this kind of proteins anchoring the heterochromatin region to lamina. But this attractive range is about 10 nanometer or so. This is very short range because that the nucleus has 10 micrometer diameter. And the difficulty is that the system is far from ergodic. During the interface, chromosomes, each individual chromosome, the genome is soft, fluid, but it's not steered very much, not in equilibrium. It's like nearly the glassy-like state. It's a very dense liquid state, probably. So, during the interface, individual chromosomes only moves a few micrometer, much shorter than the diameter, 10 micrometer diameter of the nucleus, so that the chromosome far from the nucleosome cannot reach the interaction region. So how the proper conformation like this is achieved with that kind of non-equilibrium process is a mystery. So I feel that the different hypothesis is necessary. An alternative hypothesis proposed was a non-equilibrium active force. Because the more reactions take place in type A regions, transcription and replications, so that the released free energy after the reactions should move type A domains more, vigorously. So we have the inhomogeneous distribution of the movement distributions. So that kind of active force should bring the type A region inside the nucleus. This is one possible mechanism because if the things are in the, around the center of the nucleus, it can have the more volume to move around than, than by accumulated near the nuclear periphery. So, to facilitate this large movement, type A domains would go into the center of the nucleus. This is one hypothesis. This is a paper from the India group, and this is from MIT group. But I feel the different things, I mean the different opinion. I should propose that things are not resolved yet. How many minutes do I have? 13. Okay. This is...
no, don't worry.
All right. Okay. And this is the distribution of the mean square displacement of individual nucleosomes as we saw in the previous slides. For this is, in this case, RPE1 cells, the human cells. And we have the several components. In this case, we have 3 components, actually. And this is the wild type usual cells. And this is the cells treated by alpha amanitin. The alpha amanitin is a reagent to inhibit RNA polymerase II action. So by inhibiting transcription, movement is enhanced. This is rather different from this perspective. I mean, this is rather unexpected. By stopping the transcription, the movement is enhanced. So this is a hypothetical view. So in a transcriptional complex, this is a complex of the enhancer and promoter and the mediator factors and RNA polymer. So this would form like a liquid droplet-like complex. So with this, around this complex, chromatin movement would be, tethered and constrained. And by inactivating RNA polymer, that these chromatin chains will start to, leave from this transcription complex, then that movement will be enhanced. So things are not so simple. I mean, because that interactions are so dense and the functional complex are formed in various ways. So in that situations, that the free energy released by the reaction should be dissipate into the atomic scales in milliseconds or seconds. I mean, not the micrometer scale, minutes or hour scale activation movements will not be maintained, but like in a very, vacuum-like system. But in these dense interactions, so atomic, the chemical reactions of 10 kBt or 20 kBt will quickly dissipate surrounding the atomic region. So we assumed the effective temperature would be better to use than the assumption of the break of the detailed balance or something like that. And effective temperature would be rather homogeneous throughout the nucleus. That this is probably that there are some inhomogeneous distribution of the effective temperature depending on the frequency of the chemical reactions in the system, but this would be a first step approximation. So the effective temperature would be different from the outside the body because we have the 37 degrees Celsius inside the body, but this would be homogeneous in the nucleus. That is the starting point of our modeling. So we cannot, refuse this hypothesis at present, but we started from the different hypotheses. So we have these questions. So to answer these questions, we first started from the question how domains are formed as an elementary structure block of genome. And to do that, we develop a polymer model. We assume the 8 or 30 polymer chains in a periodic box. And each chain is 500 kilobase length. And each chain is a bed and spring polymer chain, and each bed corresponds to 1 kilobase segment. You mean that each chain has 500 beads, and these chains gather in a periodic box. And 1 kilobase means that approximately that each nucleosome corresponds to 200 base, so it corresponds to several nucleosomes. So several nucleosomes are regarded to be a point, a bed in this coarse-grained model, super coarse-grained. But we will show later the super coarse-grained model. Anyway, so we considered some physically plausible assumptions to construct this polymer model. One is the dispersion between nucleosomes. This is just the exclusive volume. And the other is attractive interactions coming from this type of interactions or HP1 mediated bridging between two nucleosomes. Both HP1 mediated interactions or this nucleosome interaction take place stochastically, depending on the configuration or the binding of the HP1. So we, simulated this turn on and turn off of this attractive force in a stochastic way at each simulation step of Langevin dynamics. So this is a kind of the combination of the Monte Carlo and the Langevin dynamics simulation. So in each Langevin dynamic step, we checked this stochastic on and off trials to assume the stochastic attractive interaction between beads. And next, this would be an important point that we assume the cohesion movement along the chain. So cohesion is this. Bundle the chromatin chain. So this is schematically represented that cohesion binds here and here. In physical space, this and this position must be very near. So we assume the spring, effective spring between these two sides. And we assume that these points diffuse along the chromatin chain with the diffusion constant, d loop, and some biased diffusion with nu by consuming ATP. We do not know this is very big or very small, so that the people in the MIT group assume that this is very large, much larger than the RNA poly2, but that is not sure. I mean, so we assume we examine the wide range for D and nu, and sometimes two loops crossing each other with the probability k. So this cohesion movement was tried also at every step of Langevin dynamics simulations. Yes? Right. It is a polymer chain like this.
So you're also, so when you're saying that your extrusion velocity is V loop, so you're eliminating the part of the springs during that process, or what is that?
You mean elementary strength of string?
Yeah.
We define that this keeps the distance observed in the microscopy with some temperature definition. Spring here. Spring here, we also have some candidate range, so we examine the various cases for these strengths. But this comes from the interactions like this. So this will not be so strong as a covalent bond. I mean, so this is a few times larger than the thermal energy or something like that.
No, my question was related to this chromatin. So when this cohesion is traveling with velocity v2, you are eliminating a part of the chromatin fiber because of the cohesion, or how you are modeling it?
Cohesion
The
is
action of the
modeled by a
cohesion, yeah.
spring, moving spring.
Okay.
And we simulated the type A and type B domains with this model. And this is a high-shape pattern for type B domain and type A domain. We found many intra-domain peaks, contact frequencies in type A domain. And we have rather homogeneous contact distribution in type B domain. So we assumed this is probably due to the cohesion movement is blocked by RNA pole 2 or MCM complex. That is the complex responsible for replication. But because we have infrequent RNA pole 2 distribution in type B domain, so that cohesion moves rather homogeneously, so this gives a homogeneous contact pattern. But in this case, cohesion is trapped at the functional complexes. That loops, trapped loops at everywhere. So that gives very, intense local contact frequencies. So this is our assumption. So in this model, we assume the roadblocks in type A domains. This is a more precise mapping of the, this is micro C, it's a kind of high resolution high C. So we have these peaks, we can correspond these peaks to these ChIP-seq data or the gene region representation showing that these intense peaks appear at the CTF binding sites or promoter sites or enhancer sites or intron sites. Probably RNP2 is posed at the intron sites for the splicing or the terminator sites or the gene ending sites. So these specific sites for transcription is responsible for this enhanced contact pattern. So we assumed the roadblocks traps cohesion movement to generate these patterns. This is rather still in argument. I mean that traditionally the people think that cohesion is loaded at the transcriptional sites by so the cohesion loader protein is recruited by RNA pole 2 or something like that. This is one assumption. But the recent Hi-C measurements show that by depleting CTCF is a strong binder of cohesion. By decreasing CTCF, cohesion moves rather freely along the chromatin chain and then newly accumulates at the transcriptional site. So it shows that cohesion accumulates around the CTCF and accumulates around the transcriptional sites are the same species. So this is the roadblock picture. And so by assuming these roadblocks, we simulated the type A and type B domains. And with roadblocks, so these, transiently excluded loops are trapped at the roadblocks. So type A chains looks like, chain of loops. So this behaves like a Rouse-like chain. So the chain configuration is extended, open, and dynamically moving. But in type B domains, cohesion sweeps along the chain. That brings about the effective attraction at everywhere in the chain because the cohesion bundles the positions. So it brings about the effective attractive force. So that compactifies the type B domains. And this is consistent with the microscopic measurement. This is the gene-active chromatin domains, and this is gene-inactive chromatin domain. This is collapsed. And the size here is measured as a function of the length, sequence length of the observation. Observation window length is plotted, and the measured radius of gyration is plotted. This is for type A domains fitted to exponent of 0.4 or something like that. But the type B domains cannot be fitted with the single exponent, but show the saturation behavior. And the simulation reproduces this tendency with the exponent 0.4 for type A domain and saturated non-exponential radius of gyration for type B domains. We should say that the naive two-body attractive force between nucleosome cannot explain this saturating behavior. Because that if we mix multiple chains in a dense box, then that attractive force works among different chains. So that cannot reproduce, this saturation behavior, but the things changes in a gradual way as the observation scale is changed with that. But with the cohesion attractive effective attraction, that works only the intra-domain sites. So that collapses each chain, brings about this saturation. So this, I think this is, this has fun to see that the functions determines the chromatin physical properties. Of course, that the open chromatin, transcription factors can access DNA in this open chromatin. So physical properties of chromatin regulate functions. So functions regulate structure, and structure regulate functions. So we have some kind of the consistency or feedback relations, I guess. This is just a rough way of saying, but this is one way of argument, which is the cause and which is the result. But we should say that both are possible. And this is the snapshot of the simulated structure. And when type B domains are confined in a box with a small low density, it aggregates because of the HP1 mediated intrachain attractive force. But if the system is dense as in vivo situation, then that we have different situations. There is no further aggregation here. This can be plotted as to derive coarse-grained interaction potential between 100 kilobase scale chromatin domains. So starting from the results of the 1 kilobase resolution polymer model, then from this result, we derived coarse-grained potentials by calculating the potential mean force in a sense. So this is the result for the type A domains. So type A domain shows a mild Gaussian-like repulsive potential for different chromatin density. This is because that type A domains extends having open structures so that they can overlap. So this is the reason for the mild Gaussian-like repulsion, like usual polymer solution. But this is the case for the type B domains, without cohesion and with cohesion. And without cohesion and in low temperature, it shows attractive potential. This is the distance between 200 kilobase chromatin domains. This shows attractive interaction between domains, coarse-grained attractive interaction domains when chromatin density is low to show the aggregation like this. But when chromatin density is as high as in vivo or situation, then we have a slight repulsive interaction between type B domains. But with cohesion, the repulsion is quite evident. We have a hard repulsion between type B domains. So this is rather unexpected. Even with the attractive HP1 mediated interactions, and people use this assumption, this attractive force as a basic starting point for their modeling. But we saw the rather different, the harder repulsive interactions between chromatin domains. This is because type B domains are compactified by cohesive movement, so that some free energy cost is required to extend this compactified type B domains to start the interchain interaction between, HP through the HPR-mediated attractive interaction. So this is the reason why we have this attractive force. So, as well known in the soft matter physics, coarse-grained potentials depend on the situation. I mean, they depend on the temperature and the density and pressure and so on. So this is the difficulty to develop the coarse-grained potentials. There is a good tradition in the soft matter physics, but this was ignored in the chromatin biophysics. So we can say that in physiological density, type B domains should show repulsive interactions. Of course, this depends on the particular situation, but basically we start from this assumption. So this is the calculated effective repulsive interaction. So type A for the mild repulsion and type B, the harder repulsion. And we use the mathematically convenient potential form to represent this difference. And we assume that BA interactions are the average between them. And with this parameter setting, the so-called Flory-Huggins parameter for the phase separation is always 0. So this is the parameter usually used to simulate the phase separation. This is a mean field argument, but in many cases, this is good enough to predict whether the phase separation takes place or not. So because this is 0, this system, this particles interacting with this potentials will not show the phase separation in a usual manner. But we can see that the phase separation really takes place through the entropic reason. We can show this by using a toy model for the polymer blend system. So here that 20 type A, 100 chains, and each chain consists of 20 type A beads, and 100 other chains consisting of 20 type B beads are mixed in a periodic box. And starting from the random configuration, it shows phase separation, laminar-like phase separation. And it is interesting to see that if we confine the system into a rigid spherical wall, then that starting from the random configuration, it shows phase separation, but type B beads accumulates around the sphere surface, like accumulation of heterochromatin near the nuclear surface. So this spontaneously happens because this phase separation is induced because the type A repulsion is soft. So type A, so the larger movement of type A domains gain entropy. So it aggregates each other. Type A compartment is formed at the center of this sphere to acquire the larger space for the movement. So this is some kind of entropic reason that's somehow similar to the Asakura-Osa's depletion force.
In the phase-separated domain, the blue or yellow, do they have any other structures, or they are liquid-like, or what?
I mean, this is a toy model.
Okay.
We have the type B bit and type A bit. So this is a point, connected by springs in this case.
So within the phase-separated domain, are there any structures, or they just as random as in within the phase-separated state, like in the blue domain, they are any different than the yellow domain, or are there any structural difference between the two domains?
That's a good question. So we can see the difference in the particle distribution and particle vibration. And we are developing the mean field theory for this. And that shows that particle vibrations is renormalized into the Flory-Hagens chi-parameter. That should cause the phase separation.
And in your spherical confinement, how is the confinement interaction? How do you take the confinement?
Because that the type A beads acquire the volume for easier to move at the center. So it is rather, the movement is constrained near the wall, so that immovable type B domains accumulate near the wall, and the more movable type A domains gather in the center to acquire the larger movement, the larger entropy. I think that we should show the mean field like analysis, but it is still developing. But this simulation clearly shows the robust stable phase separation with this potential. So anyway, that we assumed this mechanism can explain the genome organization. And we identified the type A and type B domains by looking at this near diagonal region of the Hi-C contact map. If the local contacts are frequent, then we can say this is type A domain. And if the local contacts are infrequent, then we can assign that part as type B domain. This was done in genome-wide statistics. So this is a Hi-C contact map. This is a diagonal line. So this is type A domain, and this is type B domain, like this. This is renormalized by some distant factor. Distant part is enhanced. And this is the eigenvector of the largest eigenvalue of the Hi-C contact matrix. Principal component, the first principal component. This sign of the PC1 vector defines the compartment in the usual way. So with this definition, this is type A compartment, and this is type B compartment. So local contact density just agrees with this PC1 analysis. So for PC1 analysis, we need the entire genome matrix of this size. But to look at the local contact frequency, we only need this input. So the difference is 10 to the power of 7 or something like that. So by using the much smaller amount of input information to assign A, which is A and which is B, we started the simulation. Like this, we can annotate the sequence. Each 100 kilobase region is assigned as A, B, or U. U is the intermediate. So we did it in a quantitative way by measuring the local contact density. And by calculating the z-score of this density, we assigned A, B, and U region for different human cells. Different human cells have somehow different ABU sequence because of the different gene activity in these cells. Of course, that we can use the different sequence annotation using the ChIP-seq data for the histone marks, for example. So we can assign the H3K9 methylated region as type B and H3K9 acetylated region as type A. But we try to do that with the machine learning method. But we have some exceptional unclassified regions in that method, particularly that H3K27 methylation plays a dual role for the gene active and gene inactive regions. So we gave up that assignment from the histone marks, but used this assignment through the local contact intensity. But anyway, this is our test by using this data as input and try to see whether the calculated results explain the entire genome structure or not. And important point is that interface genome is not in equilibrium. So chromosomes, each chromosomes do not move much during the cell cycle. So we started, so it means that the interface chromosomes, retain keep the, starting memory of the initial conditions of the interface. So we carefully prepare the initial conditions by simulating the transition from the mitotic phase to interface by putting condensed chromosomes toward one direction. Then, putting chromosome into one direction, and then we assume that condensing disappears at this stage, and the chromosomes started to expand, and fine-graining these condensed chromatin chains into 100 kilobase resolution, and this is a example trajectory. So genome expands. We assume the nuclear membrane surrounding this, and assuming that surface tension of the nuclear membrane and pressure coming from the heterogeneous distributed repulsion among chromatin is balanced, then we reach the stationary state. And we can see that with this stationary state, that I mean that this is a simulation for the interface nucleus, we can see that genome is phase separated into yellow region, this is compartment A, and blue region, this is compartment B, and U assigned region can be found at the interface between A and B, and green here is a nucleolus. We simulated the nucleolus by an assembly of the small particles in this model, and by assuming that some attractive force between these nucleolus particles to form the liquid-like droplets of nucleolus. And this is the expansion of the one particular chromosome picked up from this system. Anyway, we can see that the yellow regions tend to merge together, or blue regions tend to merge together in this expansion process. That is the mechanism. We have the phase separation in this non-equilibrium process through the heterogeneous repulsion force. And this is individual chromosomes, and we found compartments spreading among different chromosomes. And in these simulations, chromosomes do not mix greatly because the calculation starts from the condensed individual chromosomes. I mean, in people usually started from the random configuration, so then chromatin chains easily mix. So it is difficult to explain the chromatin territories, but in this case, it is natural to find the chromatin territory distribution observed like this. This is an experimental observation. And this is the distribution of the number of the nucleoli for the simulated and experimentally observed data. And this is the radius of gyration of each chromosome, or this is the position, the distance from the center of the each chromosome. And this shows agreement with experiment. The position is somehow deviating, but we have the correlation function of 0.67 or something like that. I mean that experimental observation is somehow, it is not well defined because that it is measured from the two-dimensional image. So really that we need to compare the three-dimensional distribution. But anyway, we have a reasonable result for this. And this is the comparison for the experimental and the simulated high-C contact map. It shows good agreement for the checkerboard patterns and the domain distribution. And so this is a PC1, principal component 1 vector, shows a good agreement between simulations and calculations. And this is the genome-wide comparison for the PC1 compartment index. And it again shows a good agreement for different cells. I should emphasize that these simulations are carried out with the same parameter for different cells. So just the sequence annotation is different for different cells. So we need not to tune the parameters for different cells. And this is the averaged contact intensity plotted against the sequence separation. Again, it shows the good agreement for the simulated and observed data. And this is the high-C contact matrix between different chromosomes, chromosome 16 and 17 in this case for the simulated and observed case. And this also shows good agreement. I mean that some statistical procedure was defined, calculating the correlation function to emphasize the signal-to-noise ratio here. But anyway, the simulated data well explains the observed data. So we can say that high-C data was nicely reproduced with this assumption of heterogeneous repulsive force-driven phase separation. And in the simulated results, type B domains are accumulating near the nuclear membrane, and type A domains depleted from the nuclear membrane or nuclear lamina. Nuclear lamina is just inside the nuclear membrane. And that frequency is called the lamina association. And the similar things happens around the nucleolus, and that frequency is called the nucleolus association. This is a quantitative plot, quantitative comparison between the experiment and simulations for the lamina association along the sequence. And this is the comparison for the nucleolus association along the sequence, and shows a good agreement. This is genome-wide comparison, showing good agreement. So in this case, we did not assume any tethering factor between type B domains and LAD, nuclear lamina or nucleolus. But this lamina association or nucleolus association was induced in a spontaneous way through the phase separation mechanism. So we propose that lamina tethering or nucleolus tethering is just stabilizing the chromatin there that travels along the micrometers to come there. But when they reach there, that was stabilized by tethering factor. But formation of the lamina association or nuclear association is not due to this short-range protein complex, but coming from the phase separation mechanism. It's our proposal. And this is the whole 23 chromosomes case for lamina association and nucleolus association. So this is one summary. And we say that the model quantitatively reproduced the observed genome structure with high precision, without using this vast amount of experimental input. Only 10 physical parameters were required, and sequence annotation was used. So, and our main proposals is unfolding of chromosomes are essential to explain the genome structure. And through the heterogeneous repulsive interactions, genome was formed through the phase separation mechanism. And that mechanism explains the chromatin territories, compartments, lamina association, and nucleolus association. And this heterogeneous repulsive force came from the heterogeneous distribution of roadblocks to cohesive movement. So local functional difference generates this genome structure in this simulation model. And further, that heterogeneous movement, that is the driving force for the phase separation, remains after the system reaches the interface. So this is a plot for this is the sequence, and this is the time steps, and the dots are plotted, and this position in sequence come close to the lamina. So this shows this is the attachment, and this is attachment 2, and this is somehow the sliding of the chromatin along the nuclear lamina, and this is detachment. So that chromatin lamina association is very dynamically fluctuating, and it can be observed through the single cell measurement. And inside the nucleus, we have a heterogeneous distribution of the movement. This is the plot of the calculated mean square displacement for 4 seconds. And this should correspond to the MSD, mean square displacement, measured by my Shima's group. So that movement is slow to the nuclear periphery or nucleolus. And we see that this long wavelengths, hydrodynamical-like collective movement of 100 kilobase model domains. And this is an open question whether this kind of the long wavelengths movement of genome is related to the functional distribution in the nucleus. Anyway, so we have the whole genome model or the chromatin domain, chromatin model, and by thinking about hierarchical different levels of the models from the atomic to mesoscopic soft matter model, we can discuss how the soft matter system is designed heterogeneously through the evolution in atomic design to realize the functional. This should function. Each function corresponds to the complexes in each domain. So the whole logic should be an interesting things we are now facing, and I think there are lots of things are open to others. And this is the last slide. And most of the calculations were done by this Shin Fujishiro. And Ashwin Selvarjan analyzed the statistical data of the single nucleosome dynamics. So thank you very much.
All right. Let's thank